ETM Hotel, Rennie Sean, welcome and peace. My name is Sean. I'd like to thank everybody for tuning into this episode of Science with Sean. Please like the show, share the show, tell a friend to tell a friend that we're actually live. This build is going to be a very interesting build for the sake of what we're dealing with, the subject matter at hand. As you can see, it says, did a virus play a major role in our evolution? And we're gonna analyze that, and we're gonna see if that is true. We always talk about how viruses have been around for so long, right? They've out. Of course, they're going to outlive humans, but they've been here since the early inception, and uh, we know that they've plagued the earth for many millions and billions of eons of years, or whatever you want to say, right? But what effect do viruses have on humans outside of just infecting us and running us through the ringer and so on and so forth? Now, I do understand that before we get going, I do understand that there's a lot of people live right now. Potentially, I don't know. There's a lot of entertainment sites that are nonstop been streaming ever since 1:15 or 1:20 p.m. Central Time at the breaking news of young Dolph, being gunned down at a cookie shop in his own hood or area, at a place that he's known for going when he's in the city and getting his cookies. And people have spotted him before he arrived at the last place he would be seen alive. And so I don't know to what effect it had on him or what internal beef or anything like that. I do want to, you know what I'm saying, say it live, you know what I mean? When you lose someone in that regard, you wanna kind of deal with it. Hold on 1 second for me. I'm trying to share. By the way, I got a message from Mendel Singer, the doctor that I was talking about the other day, and I was just reading that real quick too. Peace to brother Kevin, new subscriber. Appreciate you subscribing. Like the show, share the show. We finna cook. I promise you, we finna cook. But anyway, you know what I'm saying? It's a sad story about the brother Young Dolph. You know what I mean? Loses his life. Another famous artist gunned down. We don't know the reasons. We can't speculate. It's none of our business. We don't know if this was out of a business move anyway. But we don't need to speculate or retweet, reshare, or instigate a lot of misinformation that's going around. This is the time for us to sit back and be scientific in this endeavor and let whatever evidence come out, and then we move forward from there. We shouldn't be involved in speculations and instigations and he say, she say stuff. This is a time where our people specifically, especially with social media, can create friction that ain't even really the truth. Similar to the situation where they said Yo Gotti's mother's restaurant was shot up. That wasn't the case. The restaurant didn't open till 5. However, because of Facebook rumors, tweets, and things of that nature, and fear of retaliation for something that people haven't deemed Gotti would be responsible for, Memphis police, went to the restaurant to protect and defend that. So we're probably going to see this for a while until the Memphis Police Department get a hold on it and how dirty they been over the years. You really never know. So it's unfortunate. This whole situation is unfortunate. I really didn't listen to a lot of his music, just only a couple of songs that I knew. But I know my brother, you know what I'm saying, was a fan of the boy. So, you know, it's a sad story. Anyway, man, come on, man. Like the show, share the show. Let's get these people in the building. Let's see what we can learn today. So again, the show is, we want to know what role do viruses play in human evolution. You know, you've heard me, if you've got my book, you heard me probably mention it in the book. Not this in particular, but just some viruses roles. You know, there's this thing, it's called that, well, I can't say a thing. There's it's a list of viruses that they call therapeutic viruses. These viruses once plagued and caused pandemics and things of that nature. They plagued the earth. However, these therapeutic viruses, which once upon a time was just knocking people off left and right, turned around and became what they call double agents. And you're going to see that mentioned in this particular bill. And I ain't gonna lie, I'm gonna tell you right now about that, the therapeutic viruses. So let's get to it, man. I think we got enough people in here. I don't expect a whole lot. I know people like gossip and drama, so I don't know if I'm gonna be able to give y'all some of that when it comes to viruses gang banging on one another. So hopefully everybody should be able to see the screen. Again, if you're on your, hold on. Oh yeah, I can leave my emblem up. If you are on your cell phone, adjust your quality. If you're catching the replay, adjust your quality. So you can make sure, I'm just trying to make sure that you are able to review the presentation in its full capacity and not be limited to something if you want to be able to see what's on the screen. All right, so let's go. This is our outline for today's discussion. Of course, I'm going to give you an intro, research questions, hypothesis, the argument, conclusion, and sources. So since the beginning of mankind, our evolutionary process has been linked to what some call your inner fish. Many creationists have refused this position, opting to believe in some unnatural occurrence, having command and agency over our natural world. Science begs to differ, meant to be differ, and continues to provide clues to answer important questions. By studying viruses, science has begun to understand the building blocks of life, or so it says. Today, we will set out to answer questions about our existence and the role a particular virus plays in it. Research questions: what viruses paved the way for our existence, or what virus did this virus or viruses lead to a gene being created? And do vaccines threaten our existence from a reproductive standpoint? All valid. So viruses must have played some sort of role. Here's my hypothesis, okay? It's kind of weird that I'm having to do this way, but I was trying to fake the funk. And I'm going to show you in here on how I was trying to fake the funk, because studying viruses and and especially when I have to put it in a book form and all of those things, I actually should have put this together a long time ago. But this was for fun, so let's get to it. Viruses must have played some sort of role in our evolution based on our understanding of how viruses behave. Knowing that viruses need a host, infect, adapt, and potentially mutate probably played a role in human evolution. Some viruses are more helpful than harmful, according to some scientific information that exists that says so. So I am of the belief that this could be true. Do not take this last part seriously. I am acting. Viruses have to be helpful due to our viral intake, but most importantly, our ability to adapt to certain viruses that once plagued mankind. When I said this last part was the sentence before I read to the end. Now, virus host partnerships, including genomic fusions, are commoner than many biologists realize and play an important role in underestimated role in evolution. The rise of the mammals may feel like a familiar tale, but there's a twist you likely don't know about. If it wasn't for a virus, it might not have happened at all. I want y'all to think about that. If it wasn't for a virus, it might not have happened at all. One of the few survivors of the asteroid impact 65 million years ago was a small, furry, shrew-like creature that lived in underground burrows and only ventured out at night when predators weren't active. The critter, already the product of some 100 million years of evolution, looked like a modern mammal with a body, hair and mammary glands, except for one tiny detail. According to a recent genetic study, it didn't have a placenta, and its kind might never have evolved, one, if not for a chance encountered with a retrovirus. I'm slowing this down because I'm answering research questions, but I should also be opening your eyes at this point. And its kind might never have evolved. One, I'm going to say it again. One, if not for a chance encounter with a retrovirus. Okay? So now we know that a retrovirus had to have some significance with a particular mammal 65 million years ago that didn't have a placenta. Those are the key points that I want you to take away from this. Let me continue. Unlike most viruses, which infect, replicate, and then leave their host, retroviruses elbow their way in their host genome, where they are copied and passed on to daughter cells for the life of the host. This retrovirus, however, managed to sneak its way into one of our ancestors' sperm or egg cells, able to be passed on to every cell in every subsequent generation. Virus and host had become one. Retroviruses have been infecting vertebrates for over 450 million years. Because of retroviral DNA integrates into a host cell DNA, the vertebrate genome is littered with remnants of these infections. Some of this retroviral DNA has been co-opted by the cell for a variety of beneficial purposes, such as development of placenta and the control of birth timing. The viral DNA used its own genes to copy itself, inserting those copies elsewhere in the host genome. These copies could be expressed in different parts of the body at different points in time in symbiotic relationship that gave the shrew some extra raw materials with which to develop new functions. Viral proteins already have functions. It's much easier to borrow these than to evolve them from scratch, says Aris Katzorkis, an evolutionary biologist at the University of Oxford. In the would-be mammal living 160 million years ago, a symbiotic retrovirus enabled it to evolve a placenta over many generations in order to let a fetus mature inside a mother's uterus. An animal needed a way to provide oxygen and nutrients while removing waste and keeping both blood supplies separate. So, ladies and gentlemen, this symbiotic retrovirus enabled a mammal to evolve a placenta over many generations. In this evolutionary process, it allowed the fetus to mature inside the mother. And it also allowed oxygen and nutrients to get there. And it also helped with waste. Removing waste and keeping both blood supplies separate. I don't think y'all understand what I'm saying right now. So I'm going to continue until you catch heed to what I'm saying. A symbiotic retrovirus is responsible for mammals developing a placenta. There was no placenta in these mammals earlier on. Didn't exist. Early mammals used the spare viral parts left in the junk drawers of the genome to use a viral gene to help create the placenta. And other symbiotic viruses help turn us from a ball of cells into a fully formed, squalling infant and protect us from pathogens. I'm going to run that back for you. Early mammals used the spare viral parts left in the junk drawers of the genome to use a viral gene to help create the placenta. And other symbiotic viruses help turn us from a ball of cells into a fully formed, squalling infant and protect us from pathogens. Scientists are discovering that the so-called junk DNA, a significant portion of which is at symbiotic viruses, is actually a potent force in the evolution of new species. Although the evolution of pregnancy via the placenta might be some of the most persuasive evidence that the viruses stashed deep within the genome can help give rise to new species, it's not the only proof. New studies revealing the role of endogenous viruses in more recent evolution of humans show that these snippets of DNA are helping to blur the boundary between human and virus. Humans are, in a very real sense, part virus. The boundaries between organisms are a bit more merged now, a bit more shadowy. We need to break down those boundaries, says University of Queensland virologist Paul Young. The more we look, the more we find overlap. A wonderful example of how viruses can benefit their host is revealed by the protein called Synctin-1, which is present in the placenta of nearly all mammals. Synctin-1 is required for the development of the syncytiotrophoblasts, the layer of the cells that separates the maternal and fetal bloodstreams. The Synctin-1 was originally a retroviral envelope glycoprotein gene. Ding, 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 ding. That was exapted by the host for other purposes. Another example of benefits of viral infection comes from a study of corticotropin releases hormone CRH, what they call it, which is produced in the placenta and is thought to regulate the timing of the birth. So CRH is thought to regulate the timing of the birth. You heard me mention it a few slides ago in a quote from Vincent Racaniello talking about the timing of the birth. All right. So now we have discovered that the Synctin-1 gene, and they give it another name, but this evolves from a retrovirus. So now that we see these things transpire. Let's move forward. Examinations of the human CRH gene revealed a retroviral insertion 2,000 base pairs upstream. There's a picture that I left out, and I should have put it in here for the credit and all of that. But this insertion caused by an ancient retrovirus called the Th1b, really what it was basically a fetus, the image. That's why I left it out. Infected our ancestors 50 million years ago consists of a viral long terminal repeat, the LTR. That's what they call LTR, long terminal repeat, a part of the genome that controls initiation and termination of an mRNA synthesis. Analysis of mRNA suggests that this retroviral sequence functions not as an initiator or transcription, but as an enhancer. The protein synctin, which is essential for formation of the placenta, originally came to the genome of our ancestors, and those are other mammals, via retrovirus infection. I'm going to say it again. The protein synctin, which is essential for the formation of the placenta, originally came to the genome of our ancestors and those of other mammals via a retrovirus. Placental structures have also developed in non-mammalian vertebrates. The Mabua lizard, which emerged 25 million years ago, has a placenta very much like those in mammals, and its development was likely driven by a capture of a retroviral gene. The genetic material of retroviruses is in RNA, but during infection, it is converted to DNA, which then integrates into the chromosomes of a cell. If the infected cell happens to be a germ cell, then the viral DNA, now called an endogenous retrovirus, becomes a permanent part of the animal and its offspring. One of the retroviral genes, ENV, remember that, ENV, encoding the viral glycoprotein, has been repurposed by mammals to become the synctin genes. These encoded fusogenic proteins that are involved in the formation by cell-cell fusion of the synctin trophoblast. So what are we saying here? The protein synctin, which is essential in the formation of the placenta. So without synctin, I might be saying it wrong, synctin, you don't have the formation of the placenta, because the protein synctin is essential in the formation of the protein. However, there was other retroviruses that were present to help everything else formulate. Okay, now that's where we are right now. Okay, now one of the retroviral genes is called ENV. Synctins are present in placental mammals, leading to the hypothesis that retroviral infection was driven by the evolution of placental mammals from egg-laying species. I'm gonna run this, well, I'm not gonna run it back. What I'm gonna do is I have a quote at the end regarding synctin in this very first line here, okay? I'm gonna say it again for everybody who's trying to catch up. Synctins are present in all placental mammals, leading to the hypothesis that retroviral infection has driven the evolution of placental mammals from egg-laying species. Placentas are not limited to mammals. They emerge in other vertebrates, including in certain lizards in the family of Synctidae. In one genus, Mabua, which was pictured, which is that little lizard, the placenta is structurally similar to the mammalian placenta. Examinations of placenta RNA from the Mabua lizard reveal the presence of transcripts from retroviral-like genes, like Eve genes, the ENV, that are part of the ancient retroviral genomes integrated in the lizard genome. The encoded proteins are synthesized in the placenta and are fusogenic. They can cause fusion cells, and they are which they are produced. The receptor for the lizard synctins was identified and shown to be also present in the placenta. I don't know why it takes me right now. All right, so here we go. Notice how many, every slide I'm mentioning the same stuff: placentas, synctins, placentas, synctins, retroviruses. How instrumental they are, how cohesive they are. Now, the placenta is a unique organ that mediates a complex set of mother-offspring interactions over parental investment, whereas an optimal evolutionary strategy for a mother's gene would be to allocate nutrients to each offspring to maximize her net reproductive success. Each offspring, or more specifically, the paternal genes in each offspring, would seek greater investment from the mother. Under this genetic conflict over maternal resources, an explanation for recurrent co-option of syncytins over the course of evolution is that each new syncytin gene might provide yet another opportunity for a fetus to demand more in terms of maternal nourishment. Remember that syncytin gene. So now we had a retrovirus that was key in creating, right, the syncytin gene, which is essential in what? The placenta. There you have it. Okay, so we answered a couple of research questions. Now, the occasional signatures of positive selections that are seen in some syncytin genes, including the syncytin CAR1, may further reflect this mother's offspring conflict. The extent of the genetic dissonance of interest between maternal and paternal genomes is affected by many ecological and life history traits. For instance, multiply paternity and limited paternal investment in offsprings attenuates the degree of conflict. It is possible that life history transitions between high and low levels of conflict have driven the various transitions in placentation observed today in the uterine mammals. Now, concomitant with the gain or loss of syncytin genes, thus the same retroviral genes that participate in the host-viral interaction to determine infectivity may also mediate maternal offspring genetic conflicts in mammalian genomes. That's a lot. Now, viruses can infect all organisms, from the largest blue whales to the tiniest bacteria. To a host, infections can range from unnoticed to deadly. Everybody know that. Infections can range from unnoticed to deadly. We should know that. To the virus, however, infection is an opportunity to unleash its incredible genome copying abilities. That's a fact. Within hours, it can make millions or even billions of copies of itself, using your body to replicate and make that. Now, after entering the cell, the retrovirus uses an enzyme called the reverse transcriptase. Anybody that have been paying attention in our biology class, we're dealing with transcription, reverse transcription. Remember that? So here's an example of that without getting all the other stuff stuck in our brains. After entering the cell, the retrovirus uses an enzyme called reverse transcriptase to turn its RNA into DNA. It's reversing. I'm RNA, now I'm DNA. That's reverse transcription. I came in RNA. I'm remaking myself. Now I'm DNA. Okay. Making its way to the nucleus. Once it's in, remember, when you're looking at a cell, in the nucleus of the cell is the DNA. RNA is not down there, DNA is. It would have to reverse transcript itself to become DNA. Once in the nucleus, it inserts DNA into the host genome. Most of the time when a virus integrates its genome with the host, the new hybrid genome dies when the cell and its descendants do. Sometimes, however, a virus will infect a sperm or egg cell. If fertilization occurs, the offspring will have a copy of the viral genome in every single one of its cells. It can pass the hybrid genome on to its offspring, creating what scientists call a fully endogenous retrovirus, a fancy term for a virus that comes from within. The idea that a symbiotic virus or a symbiotic relationship could have such a profound influence on the evolution of a new species is both new and controversial. Here's why. For more than a century after Charles Darwin published The Origin of Species, scientists focused on competition as evolution's chief driving force. Biologist Lynn Margulis wasn't convinced. The late University of Massachusetts researcher believed the cooperation also played a role. Her evidence lurked in every cell of every plant and animal. Beginning in the late 1960s, Margulis argued that our cells contain symbiotic bacteria known as mitochondria and chloroplasts, which earned room and board by either supplying energy or producing food from sunlight. Margellis's idea was ridiculed, and she struggled to find a journal that would publish her hypothesis. By the 1990s, however, enough genetic evidence had accumulated to show that Margellis was actually right. Symbiosis was responsible for some of the most significant evolutionary leaps in the history of the planet. Most scientists, however, view this event as anomaly, a once-off freak occurrence that, although significant, didn't play a role in the ongoing evolution of most species. Margellis, though, saw symbiosis everywhere and believed that this softer, gentler side of evolution was getting short shrift in research. Although most symbiosis research was focused on the role of microbiome, the virus tucked into our DNA can play a similar role in splitting apart two populations, turning one species into two. The first wedge scientists discovered was a protein called syncytin, turning one species into two. A team of virologists at the Fred Hutchinson Cancer Research Center in Seattle identified genes derived from the human endogenous retrovirus H, which is HERV. Remember that ERK V that I talked about, so they call it HERV-K, that were active around the time when the embryo was just 8 cells. Of the many known endogenous retroviruses in human, HERV-K is the newest. It inserted itself as recent as 200,000 years ago. It's so new that several of its copies in human genome can still produce viral protein. To prevent this, adults keep a tight control of HERV-K by switching it on and off, though this isn't the case in very young embryos. Rigi Pera and Wasaka found, but far from being detrimental, which are two of the researchers of a team, but are far from being detrimental. HERV-K activates key genes that help transform a single cell into a fully formed infant. HERV-K activates key genes that help transform a single cell into a fully formed infant. These HERV-K viral particles and proteins also help protect a tiny ball of cells from being infected by other viruses, the researchers show in a 2015 Journal of Nature paper. Excuse me. HERV-K may also have played an important role in separating some of the first humans from their primate ancestors. You don't be talking about this, where you at? You Mr. Charles Darwin and evolution, man. You don't talk about HERV-K. What's up with you, Huh? Why you ain't talking about HERV-K? HERV-K may have also played an important role in separating some of the first humans from their primate ancestors by making small adjustments in when certain genes were switched on and off, according to Rigi Pera's research. But with tens of thousands of viruses embedded in our genome, scientists have only just begun to explore their potential effects. In a recent paper in Science, University of Utah geneticist Cedric Fischel found that these viruses played a key role in evolution of mammalian immune systems, and even now continue to tell certain immune system genes when to turn on and off. These viruses put us on a fast track to evolve, and the bells and whistles needed to evade other viruses, Fischel says. These viruses are already equipped with all kinds of weapons to evade our immune system that now can be recycled. The effects of retroviruses, however, aren't always beneficial. Originally, many probably caused diseases in the organisms they infected. Look no further than the Australian koalas. But in the case of the endogenous retroviruses, We're far removed from that time. The genetic results we see today are the silver lining to the what otherwise might have been a deadly epidemic, for Schultz says. Curse of Katsourakis and Pera both believe that the endogenous retroviruses are blurring the lines between virus and human. It's changing how we think of ourselves as a species. Such an intimate interaction between ourselves and these viruses and exchanging DNA that's useful for us has really molded how we're now thinking of ourselves as a dynamic soup of DNA that's now infiltrated by viruses. We now know of two examples of how ancient retroviral infections have influenced the function of the placenta, and they are likely more to be discovered. If not for such retroviral infections, mammalian pregnancy would likely be very different. Without a placenta, human embryos might have short gestation times and be nourished in a pouch like marsupials. So instead of y'all giving birth through the womb, y'all would have been doing what marsupials do. Without retroviruses, many species might still be laying eggs, and that includes humans. Without retroviruses, many species might still be laying eggs, and that includes you women in the chat. Now, in 2016, scientists at the University of Utah found that the endogenous retroviruses in the human genome, which originally came from a virus that infected our ancestors roughly 45 to 60 million years ago, switches on a gene called AM2. This is another gene. So we had the HERV-K, we got the AM2. When it detects a molecule called interferon, which is the danger signal that warns the body that it's suffering a viral infection, the AM2 then focuses the infected cells to self-destruct to prevent the infection from spreading any further. These ancient viruses have become double agents, helping our cells to tackle other viruses that they are trying to attack us. So they just give you a little bonus right there. All that to say this. Numerous posts have falsely claimed that mRNA vaccines used against COVID-19 target a protein called Singleton-1, which is needed for placental formation and successful pregnancies. Many of these posts are also baseless, imply that vaccines will make people infertile. These claims are untrue. No available mRNA vaccine target a protein called Singleton-1. One such post reads, the mRNA vax targets a protein called Singleton-1, which is a protein contained in the CV spike protein complex. The problem is there is no mechanism to turn off or stop the destruction of Singleton-1, which is also found in the brain and female reproductive system. Therefore, Singleton-1 will continue to be scavenged in the body. Without Singleton-1, no placenta can form. The brain manifestation of no Singleton-1 are MS, Parkinson's, schizophrenia, psychopathy, and I posit, any neurodegenerative condition. The claims that these mRNA vaccines will cause the immune system to attack Singleton-1 are unfounded. Singleton-1 is not contained in the Sars Cov2 spike protein, and the Sars Cov2 spike protein and Singleton-1 are not very familiar. Singleton-1 is not part of the Sars Cov2 spike protein, y'all. There is no evidence that mRNA vaccines for COVID-19 have a negative effect on female fertility. Conclusion: symbiotic retroviruses play an important role in human evolution. However, evolution of all mammals. The protein Singleton, also known as the HERV-W1 gene or the HERV K that we saw earlier made it possible for the placenta to be what it is today. Science continues to answer questions regarding human evolution, but also the role that viruses play in our existence. We owe a huge deal to this specific virus, although many years ago it could have been responsible for killing whatever hominid who crossed its path. As far as mRNA vaccines impacting the female fertilization, that has been proven to be false, especially regarding the syncytin protein. mRNA vaccines never enter the nucleus of the cell, and today we have more evidence of the positive support mRNA vaccines provide mothers as they gain protection against a virus that has manipulated our lives over the past few years.
Welcome to the lab, everyone. Today's video is going to be a little bit different, as we usually teach anatomy from the full bodies behind me, but today we're going to teach from this amazing little fetus that has already educated millions of people. We'll talk about the story of how this came to be, how it was donated to us, and even some of the mysteries surrounding the fetus, such as gestational age, how many years this little body has been teaching people for, a lot of cool stuff, and of course, discussing the most miraculous concepts about how us humans develop. So let's do this. So before I unveil the fetus here, I do want to have this discussion or this talk about cadaveric education, and specifically the difference between teaching, say, from a fetus versus a full adult body that's lived a full life and chosen to donate their bodies to science. Yes, there's a little bit more tragedy associated with a fetus dying, or any young person for that matter, but let me give you guys a little bit of a shift in perspective, or at least our perspective on this. Justin and I get asked all the time, does it bother you guys to work with the dead, to do dissections, to remove tissues, remove organs and things of that nature? And without hesitation, we say, no, it doesn't. Because we believe that cadaver labs are much more about life than they are about death. Now, that may sound a little ironic, because when you walk into an anatomy lab, you see deceased bodies. However, the dead teach the living. And what we mean by that is that all of the thousands of students that have come through here, the millions that have been on our online platforms and learned from these bodies, can now go out and help the living live happier and healthier lives. Think about a healthcare professional going to school and learning from a full body or an adult body or even a fetus, and taking that knowledge into patient care and all the people they interact with on a day-to-day basis to, again, help them live happier and healthier lives. So the people and the families that choose to donate these bodies to science leave this priceless anatomical legacy behind, so that we can be educated to continue to help the living. So now that I got that out of the way, let's take a look at this amazing body. You ready? Here we go. So as you can see, amazing fetus here, and I want you guys to think about the gestational age, or in other words, how far along the fetus is. We're talking like 10 weeks, 20 weeks, 30 weeks, first trimester, second trimester, third trimester. You can see a very well-developed head that's starting to get more proportional with the rest of the body. You can see the ear there, and the upper limb with a well-developed hand and fingers there. Even if you look closely there, you can see some fuzz or some hair developing on the back of the head there. But before I rotate this around to show you the umbilical cord and the placenta, and even male or female, I want to tell you about how this body was donated to us. There was a high school teacher that had been teaching for over 30 years with this fetus. So over 30 years of education for high school students. That was 8 years ago when this body was donated to us. So we're knocking on the door of 40 years, maybe even 40 plus years, that we've educated thousands in our lab, millions with our online platform. This little body has done quite the educational feat. But there were two mysteries, or two pieces of information, that we weren't given when we were donated this body. One was the exact gestational age. So we were left with making our best guess based upon the developmental features. So let's keep rotating this around so you can see some of these other features. Again, you can see the arm more fully now, the umbilical cord coming into view around the leg there. And as we continue to rotate around, you can see the feet and the well-developed feet, and even the genitalia, which we're seeing male here. And if I continue to rotate around, you can see all of this tissue here, or the structure, is the placenta. And so we kind of use our best estimation based on the developmental features of the fetus to estimate that the gestational age is anywhere from about 26 to 30 weeks. Essentially kind of that transition from second to third trimester. So that means there was about 8 to 12 more weeks of development left, depending on where we fell in that 26 to 30 week period. So a lot more growth or size development. And speaking of size, let me give you some more context about the actual size of this fetus. So here's my hand. Let's show it in the frame here so you can actually see about the context. So size of my hand to the actual size of the fetus. Now, part of the reason I'm also wanting to show you this is because I'm going to show you on a real uterus, a normal size uterus, comparing it to the size of this fetus. And to give you guys an idea, about here to here, I measured before the video, is about 8 inches long. And I'll show you that comparison on the uterus in just a second. But before I do that, I just want to mention the last mystery surrounding this fetus. We don't know what happened. What I mean is we don't know what happened to mom, and we don't know the exact circumstances surrounding the death of the fetus. What we do know is that the family still chose to donate the body to science and education, which is absolutely amazing. And there's this interesting concept and discussion that we often have in this lab regarding this particular fetus. Remember, 26 to 30 weeks in gestational age. There have been plenty of preterm fetuses or preterm babies born in that 26 to 30 week period, and they've survived. It makes us wonder, because remember, this fetus is almost 40 years old. If born in 2021, would this fetus still be alive? Did we have enough advancement in medical knowledge and medical technology to do that? So again, it kind of pushes that idea of furthering our medical knowledge and pushing those boundaries to the point where can we continue to get better and even save more lives earlier in the gestational period. So now let's show you a real uterus. We actually have 2 bodies we're going to use. One, we're going to show a uterus where we reflect the abdominal wall up and look at it from above. And then we've got another body that we've cut in half. So you'll see a little bit less than half of the uterus, but you'll be able to see inside. I think you'll be surprised about the size of each uterus here. So let's take a look at the first body. So I'm going to reflect the abdominal wall here. This is the pubic bone. This is about where the 6-pack muscle would be. Up this direction would be the rib cage. I reflect that away, and you'll see this fatty tissue called the greater omentum. It's an apron of fat. All of us have it. And I'll reflect that away. And then finally, we see all the small intestines here. And weird, we've got to reflect this away too. And finally, once I reflect all that tissue away, we have exposed the uterus right here. So essentially, this is the top of the uterus right here. Some other cool structures, if I pull down here, pull this tissue up here. The uterine tube runs in this connective tissue right through here. Let me get my hand out of the way. It runs in that tissue right there. But the tube is on the top edge of that tissue here, and I'm pinching the tube with the probe there. And I can actually get to the end of the uterine tube right there. Now, sometimes it's referred to the fallopian tube, but we prefer uterine tube here. Oops, I dropped it. Picked it back up again. But then let me show you the other important structure, and that is the ovary, which produces the eggs, or the ova for eggs, or plural in the technical sense. But we'll use this to talk about the size variation when we're talking about a regular sized uterus without a fetus developing, and then how it changes during pregnancy. But let me show you the other body here. So over here, you can see this is the cut half right through here. And specifically, this is the right half of the uterus here. And you can see this is the inside. The majority of this tissue is made of smooth muscle. Over here, again, the ovary and that uterine tube, you can see embedded in that tissue right through here. Now, what's interesting about this when we're talking about fertilization, let's talk about that for a second. Here, the ovary releases the egg on about day 14. That egg then enters the uterine tube, gets in that uterine tube. Then sperm cells will travel up, enter into the uterus, go up into the uterine tube, and this is typically where fertilization takes place. That fertilized egg, or that zygote, will then descend down into the uterus and implant into the wall of the uterus. That is crazy to think about. We started as one cell. It's a relatively large cell, but one cell nonetheless, and then it develops into this, and then eventually continues to grow into this. I mean, think about that. That's crazy. And now we go a little bit further and talk about the size of the uterus. Let's talk about the regular size. So the regular size of the uterus, again, right here is the uterus on the other body. It's about 3 inches long. So where my finger would be going underneath there, that would be about 3 inches in that vertical length there. And then it's about 2 inches wide. 2 inches wide, and then 1 inch thick. So it's kind of easy to remember 3, 2, 1 as far as inches. Now, that's a uterus that's never been pregnant. When a uterus does get impregnated or start to have a developing fetus, think about the changes that have to undergo inside the uterine cells. They have to actually undergo hyperplasia and hypertrophy. In other words, the cells have to divide and also get larger to accommodate that fetus developing inside. So go back to those measurements one more time. 3, 2, 1. Approximately 3 inches long, 2 inches wide, and 1 inch thick. But those are just the outside measurements of the uterus. Remember, the fertilized egg or the zygote is going to implant on the inside lining of the uterus and then grow and expand within, again, creating quite the remarkable change in the uterus. So, everybody, thank your mom. If you are a mom, you're awesome. Thank yourself. And if you donated the sperm, good job. Thanks for watching the video, everyone. Hopefully you learned something new. And I want to take this time to say thank you for all of the support. We recently crossed 1 million subscribers on YouTube. And
there you have it. So let me, my bad. That was my bad. So there you go. And my sources. The rest of my sources, of course. Those Journal of Nature papers can be found in the sources. So if you ain't got your book, if you do not have your book, in the description of the video and also in the chat, go to what's all, go right there, hit that link, get your book today. Quit waiting. Oh, Sean, I'm gonna get it in a minute. I'm gonna get it next week. You said that 2 weeks ago. Get the book. I promise you, as a tool, this book as a tool will help you understand what we're in the middle of, but also help you correct misinformation, because your family members and friends normally get a lot of their information from other friends. Get the book. I promise you, as soon as you open the book, you're going to understand some things, and it's going to help you evolve as far as your understanding and your learning and the behaviors of viruses and things of that nature. So that concludes the presentation, man. What do y'all think about that? Of retrovirus and other retroviruses played an important role in human evolution, a major role in human evolution. Without a retrovirus, you wouldn't have a placenta pretty much, right? The retrovirus being the cause of the gene that created the placenta. A retrovirus was responsible for a gene that helped create the placenta. Now, I put the fetus video at the end since we were talking about placentas and things of that nature, and we talked about fertility and how, you know, the birth and all of that. So that kind of tied everything into that. But I also had to slick in the use of trying to educate y'all on this SINCTIN1 gene, right? Because anti-vaxxers have tried to argue that there was a fertility issue. And of course, the vaccine doesn't get anywhere near the nucleus of the cell or dwell in the realm of the DNA. So how could it affect or invade in that? So, if you understood viruses and how they act and behave and things of that nature, then you understand the gist of everything here. Any comments, concerns, likes, dislikes, contentions, anything like that, please don't hit me with no whatever Unc just sent, because I ain't even dealing with that type of a person today. Any questions regarding the presentation, specifically Symptom 1, or HERV K, or ERV, whatever you want to call it, those specific things that were influential in the evolution of the placenta, or the mammal that was so freaking old that didn't have a placenta. Imagine that, though. Imagine that thought process, that if this key gene didn't evolve, right? If this didn't evolve, Benita, you would have been giving birth. You would have been laying eggs. Your children, you would have had to sit on your children and keep them warm. As a human being, think about that. You, right now, think about that. To all the women in the chat, if you've had kids or you plan on having them in the future or whatnot, whatever the case may be, I totally understand. How would you keep your egg warm enough for a long period of time and then know when it's going to hatch? You're not a bird. You don't know nothing about that. You know what I'm saying? How would you know when? Now, eventually, through evolution of the brain and all of that, humans would naturally determine how long that would be before hatching. Yeah, like a chicken. Imagine you having a baby like a chicken. Yeah, I ain't gonna hold too many people though, like a couple of minutes, but I was trying to see if anyone had any questions, comments, or concerns regarding the presentation. But just imagine that. Imagine, fellas, let's put it in today's terms. You got a baby on the way, she pregnant, and all of a sudden she lay an egg, bruh. What you gonna do with that? She just laid a 2-foot egg, bruh. Imagine that. 2 feet, a 2x3. That's a whole lot of pushing. A 2-foot egg, bruh. A 2x3, 3 feet wide, coming up out of there. Potentially, I'm not saying, you know what I'm saying? Potentially a 2x3. And then imagine the women that have had C-sections, right? Meaning the baby was too big inside of them. Man, that's a 4x6. Man, them women would be stretched as all out get out, bruh. You'll never be in that thing. Maybe it'll take a long time, but you ain't gonna snatch back like that. He said, think of the baby showers, everybody decorating eggs. Quit laughing, Jeremy. You know it's true, bro. You know it's true. Absolutely, James. But I see y'all ain't really got no. I mean, y'all was rocking with it. For those of you who paid attention to it, man, you know what I'm saying? I was kind of rocking with it. I don't know how many of y'all knew which gene it was, or how many of y'all knew which virus played an intricate role. I mean, was there anything that we went over today that you already knew? If you already knew, put a 1 in the chat. If there was things in that you didn't know at the conclusion of the presentation, put a 2 in the chat. I know some of y'all might have to catch the replay. I understand that. But what's y'all thought process on what we just went into? Zion, you heard the gene specific? Oh, okay. I got you. Like, oh, you heard like a virus played a role in the evolution. Okay, but you didn't know which gene or if it affected the placenta and stuff like that. Okay. That's what's up. Well, I don't know. I don't know. You ain't answered that, I'm assuming. Because you said 1, 2. So I'm pretty sure you understood that a virus played a role in human evolution. But to what degree, which virus or whatever? Oh, okay, okay. You said right. All right, cool. Yeah, man. I just wanted to merge the two things because when we talk about viruses, we can rightfully talk about evolution. They're interconnected, and now we know how interconnected that they are because we don't exist without them. So I want y'all to think about this. We don't exist without retroviruses. Now, SARS-CoV-2 is not that, right? But I want you to understand the importance of viruses here. They will outlive us. They're more important than we are. And there was a quote that was told to me today. And I don't know if she knew that I was doing this, or I know she didn't know that I was doing this. And I don't know if Brother Chill knew that I was doing this, but this bags that quote up about the significance of mankind thinking that they are the only organism, the most important organism on this planet, and they're not. Because you don't exist without a retrovirus. So that's that on that. And shout out, like the brother said, about the women and their ability to be able to, well, develop a placenta, have the ability to develop a placenta. Because I don't know, y'all might be some good egg layers around here. I bet it wouldn't be. Look, peep this. It wouldn't be people who had 18 kids and all of that. Back in the day, they used to have big old families. I bet you the brother wouldn't have had no 18 kids that he's seen an egg come up out of there. With the most self-entitled, yeah, it's a fact. Appreciate that, Zion. But let me see. I was working on that for a couple of days, putting that together. I was gonna go over it again today, but I was like, I'm just gonna throw it up. Got to measure twice, cut once. I should stop there. I'm reading a couple of books at the moment. I usually just read one book, but for some reason, I've started. I started another book. And this book is dealing with the West African and African American experience, the cohesiveness of the two, and it's talking about Herskowitz and a whole lot of other things is going on. Then it's I don't know, it's kind of all over the place. My mind on science, but you know what I'm saying? It's on Africa. It's on Africa too. I don't want to get involved in the shenanigans of a conversation that just continue to be circular when people are just now starting to study a subject matter. So I'm staying all the way away from that. I'm not even engaging. I'm advancing the conversations forward. And I want to provide people with information that they could actually use utilize and help them advance. I see our kids can benefit from a presentation like this. Even, y'all can benefit from a presentation like this. So hopefully it serves its purpose. Some viruses help mankind evolve to where we are today. Facts. Facts. Retroviruses. Listen to this name. Adenoviral vector vaccine. An adenovirus. What is that? We did this before on the Moxie Warrior Clan channel where we talked about adenoviruses, right? It's are common viruses that cause a range of illnesses. They can even cause cold-like symptoms, fever, sore throat, bronchitis, pneumonia, diarrhea, and some more stuff. We'll take an adenovirus, weaken it, replicate it, and then engineer it and use it to make vaccines to fight against other viruses. Johnson & Johnson. That's an adenovirus vector vaccine. That's an adenovirus vaccine. That virus has been altered, and now it's used to fight Sars Cov2. That's in the book too. I believe if you look in the book, I don't think I go too specific, but when we talk about different stages of viruses and stuff like that, I think I get into that. I can't even remember, man. So much studying. But I know for sure we did a build on that, and Chavis even did a build on that because I made sure that he did, because we were arguing about Johnson & Johnson versus mRNA vaccines one day, just not really arguing, but having a little fun with it. So by 2022, we should have an updated information from Moderna's mRNA HIV vaccine. I think it's called the mRNA 1855 or something like that. They're doing their research and studies on that. So if they solve that, that's going to be a huge win for mankind, although it took quite some time. But let's just give y'all some little insight, some little information. Are you late, Lamar? And Jeremy said, quit calling y'all chicken heads. Some people, sorry. He said he did quit. I can't call everybody that. All right, so I just got another message from Mendel Singer. This is cool. He responds. He says, thanks. Trying to stay on top of truth on a schedule for your shows will sometimes lead to an error. You've taken care of it. Sometimes do that. No, he says, you've taken care of it. Scientists do that. Keep on. His brother's encouraging it. He's encouraging what I'm doing right now. Okay, I see what he's saying. He's on a schedule when I'm doing my shows. Yep. He said that's exactly what scientists do. He says if an error has occurred, science is self-correcting pretty much. And Let me make sure that they get it. Let me send them the shizzle. But yeah, man, I think that's dope. This dude is a scientist. He's just encouraging what I'm doing. He's telling me to go for it. And y'all ignoring it, man. So y'all wanna see. See, if somebody else would have wrote this book, man, it would have did really well. It would have did really well. But it's all right. Maybe when I transition, you know what I'm saying? People want to get the book then. Says I'm gonna be on the rewatch list. I got you. But yeah, man, get y'all a copy of What Sars Cov2 Taught Me. If I can get Mendel Singer on, I would love to get him on an interview and we can talk about the myocarditis situation. I would like to get Vince on, but the way Vince's schedule is set up, he hadn't even responded to emails per request from other doctors who requested his services. And right now he's teaching, so it's really difficult. School's back and all of that. So, man, look, I'm about to slide out of here. I appreciate everybody for tuning in. Like the show, share the show, tell a friend, man, get the likes up, get the shares up, rewatch it. It's not long. This thing gonna be about an hour and 15 minutes, really, and it's a little bit less than that. So there's a huge lesson in all of this, and I appreciate everybody for your support. And for those of you who have actually picked up your copy of What Sars Cov2 Taught Me, I appreciate you. If you haven't posted on your social media sites, please, I'm asking the same person to do this again. Please post it on your Facebook pages. Tag me in it, right? If you got the book, tag me in it and let me know.